So in chapter 4, we're going to talk about some of the anatomy of both prokaryotic and eukaryotic cells. So again, when we're talking about prokaryotic cells, we are talking about both bacteria and archaea, but we tend to focus more on the bacteria. And with eukaryotic cells, we're going to talk about not only their structure and function, but we're also going to talk about a little bit of the evolutionary history of eukaryotic cells, and we're going to get into what's called the endosymbiotic theory. This is a really classic way to compare prokaryotes and eukaryotes, and it gets pretty easy to start just listing the different features or putting them in a table versus having to write a giant paragraph. And so, in general, and of course there are exceptions to rules, etc., but in general, a prokaryote typically has a single chromosome. It is usually circular. But there are exceptions. For example, Vibrio cholera, the bacterium that causes cholera, actually has 2 chromosomes. And the genus that I did my PhD on, Bacterial, has a linear chromosome instead of a circular chromosome. There are other bacterial genera that have linear chromosomes as well. In prokaryotes, the DNA is not associated with those histone proteins. Prokaryotes don't make histones. And prokaryotes don't put their DNA in a nucleus because they don't have a nucleus. Instead, they keep it in a region called the nucleoid. Prokaryotes also lack membrane-bound organelles. Again, for the most part, there are a few species of bacteria that have what are called inclusions. So it's not necessarily like an organelle, but they do have structures within their cell that have membranes. But broadly speaking, most people will accept if you say that prokaryotes have no membrane-bound organelles. Again, for the most part, bacteria have a cell wall made out of peptidoglycan. Archaea have a cell wall made out of pseudomurin. An exception for the bacteria would be the bacterial genus Mycoplasma has no cell wall, but that's very rare. And prokaryotes, again, typically reproduce by binary fission. We can contrast that with eukaryotes, which are most often diploid, at least in some of their life stages, meaning they have paired chromosomes, and those chromosomes are usually linear. The chromosomes are tightly packed within a nucleus, and the way that they get packed up is by being wrapped around those histone proteins. Eukaryotes also have other membrane-bound organelles. For example, the mitochondria, some have chloroplasts, they have the Golgi, the ER, all of that. When eukaryotes do have a cell wall, which is not found in all eukaryotes, it is found in our photosynthetic eukaryotes. They typically have, like plants, they have a cell wall made of cellulose. Fungi have a cell wall made of chitin. So it's a polysaccharide cell wall as opposed to a protein polysaccharide mix. And they divide most frequently by mitosis. To get deeper into the prokaryotes, again, unicellular, don't have a nucleus or other membrane-bound organelles, and include the domains bacteria and archaea. So again, I don't want you to kind of fall into this trap of thinking that, well, because bacteria are single-celled, because archaea are single-celled, they are not complex. That's not true. These organisms can still be very complex, even though they might lack some of the cellular features that eukaryotic cells have. Bacteria and archaea can vary in size and shape. On average, a prokaryote is anywhere from 0.2 to 2 microns in diameter and 2 to 8 microns in length. However, there are some exceptions. For example, this bacterium right here is the largest known bacterium. It's actually 3/4 of a millimeter. So that's pretty big. And the smallest is in that genus Mycoplasma. It's that Mycoplasma genitalium, only 0.2 microns. The reason that this is really beneficial is because this gives prokaryotes what we call a high surface to volume ratio. They have relatively little volume because they're very small, but they have a lot of surface area. And this allows them to grow more quickly than a larger cell. It also allows them to do nutrient exchange more quickly and more effectively than a larger cell. We can start to differentiate some of our prokaryotes based on a number of different features, including morphology and arrangement, which we'll get into in this chapter, chemical composition, nutritional requirements, and biochemical activities, which we'll get into a little bit later. And sometimes you need a combination of all of these things in order to help you identify what species you're working with. When we're talking about morphology, morphology is the shape of an individual cell. Most bacteria have a relatively consistent shape. We call them monomorphic, meaning every member of the species will look the same. However, sometimes there are bacteria that are called pleomorphic. Different members of the species, depending on perhaps what life stage they are in, what environment they are in, may have a different morphology. So the morphology can change depending on the environment. We also can consider the arrangement of microorganisms, how they are grouped together, and that can be descriptive as well. Our three classic bacterial morphologies, and here I'm defaulting to bacteria because that's mostly what you're going to be looking at when you're looking at prokaryotes in the lab. You're not going to be really looking at any archaea. So our three classic bacterial shapes are coccus, which is this round shape that you can see in these two scanning electron micrographs. And you can see they can have different arrangements. These are arranged like a chain, whereas these up top are arranged more in clumps. You can have a bacillus shape or a rod shape. You can have longer rods like are shown here, or you can have kind of these shorter rods as shown in the bottom image. And then we have spiral, and you can have a relatively simple spiral. This might be something like Vibrio, or you can have a much more complex spiral, and this could be something like Bacterial. When we get into the archaea and talk a little bit more about them, some of them can have much more unusual shapes. I think this is probably my favorite. The genus Stella has this star shape, and then there's even this rectangular one in another genus. So again, morphology can be helpful in narrowing down what species you might be looking at, but you can't always use it as a definitive identification because there can be multiple species that have the same morphology. When we're talking about arrangement, especially when we're talking about the coccus-shaped bacterium, those can again be maybe not necessarily a true identification, but they can help you narrow it down. So you have sometimes these coccus-shaped organisms that will grow in pairs. We call that a diplococcus. You can have them as a chain, streptococcus. They can grow as a tetrad, a group of 4. They can grow here in this like kind of cool little group of 8, or they can grow in a big clump, like a staphylococcus. With the rod-shaped cells, the arrangement is not quite as interesting. You can have a single cell, you can have 2, you can have them chained together as a streptobacillus, and then you have this called a Coccobacillus, and we saw that on the slide a few slides back, where they're technically rod-shaped, but they're not like really long. They're kind of these short rods that look sort of halfway in between a coccus and a bacillus, and so we call that Coccobacillus. And with the spiral, you can have a very mild spiral, a little bit more of a spiral, and then a really tight spiral. So you can start to really narrow down what you're looking at as you start looking at the cells under the scope. And we're not going to do in the lab any staining of these cells, but you will look at prepared slides that have these morphologies. So the spiral bacteria are characterized by a helical shape, and they have the sporillum have a more rigid body, so they're not as flexible. The spirochetes are super flexible. They're actually really fun to look at under the microscope when they're living because they move so quickly. There is no like grouping or clustering of spiral-shaped bacterium. They're always going to be found individual. And now we can start to talk about the anatomy of a bacterial cell. So this is just a cartoon of a bacterial cell, and it's trying to illustrate as many features as possible. So not every bacterium is going to have all of these features, but every bacterium will have some of them. And the features that every bacterium should have are in red. All of the extra features that a cell could have are in black. You should be able to, if given an image like this, be able to label what the various parts are and what their functions might be. So again, we're going to, in the next few slides, take kind of a deep dive into each of these parts of the bacterial anatomy, but I just wanted to give you this overview and again highlight that the red are features that all bacterial cells have, whereas the black are not found in every single bacterial cell. And so we will start from the outside, and we talked a little bit about one of these structures on the outside of the cell before, but broadly, when you're talking about external structures formed by a bacterial cell, one of the first type of structures that can be very helpful is called the Glycocalyx. And what that means is it's just a sugar coat. It's a gooey, sticky protection for the cell. It can be made of polysaccharides and polypeptides kind of mixed in together. And there are two ways that the Glycocalyx can be organized. They can organize it like a capsule where it's kind of tightly packed and attached to the cell itself. And when we talked about microscopy in the previous video, we talked about how you can visualize a capsule using a particular type of stain. The other way that cells can form a glycocalyx is as a slime layer, which is exactly what it sounds like. It's kind of a sticky, slimy secretion from the cells, but it's more loosely organized and not as tightly attached to the cell itself. Oftentimes, the microbes that produce these structures use them for really two main things. One, because they're sticky and gooey, they can use them for attachment. And if they're pathogens, that means they can use them to attach to us. The other really important thing that this kind of external structure can do is actually protect the cell from the immune system. It protects the bacterium from being eaten up by cells of the immune system. And this image here is of a bacterium called Streptococcus pneumoniae, which causes pneumococcal pneumonia. There are some strains of this bacteria that produce a capsule and some that don't. The ones that do produce a capsule cause a really robust infection in people. The ones that don't produce a capsule don't. So it can be very important for a cell to make it. The other, feature is if the cells form a biofilm, like this community of microorganisms living together, it can help that biofilm to stick, but it can also prevent the cells within the biofilm from drying out. So that's pretty important as well. And a classic example of bacteria that form a biofilm using a glycocalyx are those that form the biofilms on our teeth. So especially members of the genus Streptococcus. So Streptococcus mutans is a very, very common bacterium in the mouth. And if people aren't really good about their brushing, that can lead to plaque buildup and eventually like destruction of the teeth and gums because it causes cavities.